Hi everyone, today I'll be talking about the first project of my PhD thesis, which is titled Cross-sectional Characterization of the Impact of Alpha-synuclein Spreading in a Mouse Model of Parkinson's Disease. As you all may know, Parkinson's disease is a progressive neurodegenerative disorder and is generally characterized by slowness of movement, rigidity, and tremors. And these characteristic clinical manifestations appear due to the extensive loss of dopaminergic neurons in the nigrostriatal pathway. And as shown in the image here of a coronal slice of the substantia nigra of the human brain, we see a strong staining of neuromelanin, which is this dark pigment here, which is found in dopaminergic neurons. And as you can see, this pigment is severely lacking in the brain of a patient with Parkinson's disease. And in fact, this degeneration is one of the neuropathological hallmarks of Parkinson's disease. The second being the presence of protein-containing inclusions, which are known as Lewy bodies and Lewy neurites, and these are essential for the neuropathological diagnosis of the disease. And the primary component of these protein-containing inclusions is the aggregated form of the alpha-synuclein protein. And this protein is a small protein that's highly soluble, natively unfolded, and while the precise function is not yet completely understood, it's enriched at the synaptic terminals, which suggests a role in synaptic maintenance and neurotransmitter release. And importantly, when alpha-synuclein is recruited into the pathological inclusions, it undergoes extensive phosphorylation at the serine 129 site, and this causes the conversion of the protein to its aggregated misfolded form, which is generally the toxic form found in the Lewy bodies. And so while the precise role of alpha-synuclein in disease pathogenesis remains unresolved, several studies have sought to examine its role and have shown evidence for the hypothesis that alpha-synuclein seems to be spreading transneuronally. In fact, converging lines of evidence have pointed towards misfolded alpha-synuclein propagating throughout the brain, where specifically human postmortem studies have shown evidence for endogenous alpha-synuclein aggregates appearing in transplanted cells years after transplantation, and as well as cellular models and animal models, studies in which have provided evidence for alpha-synuclein spreading from one cell to connecting cells, as well as from one region to connecting brain regions. And these studies have led to the recent hypothesis that alpha-synuclein seems to be spreading in a prion-like manner, so spreading via neuronal connections. However, there's still no direct evidence for alpha-synuclein being the cause for the degeneration and the atrophy, which give rise to the disease presentation. And so the goal of my project is to characterize the signature of network spreading and disease progression by examining atrophy using MRI in combination with traditional cellular techniques in order to examine the underlying cellular pathology. And this is done using a mouse model of Parkinson's disease, where the goal is to ultimately establish a biological model in which we can serve then to as a test bed for the development and testing of different therapeutic interventions. And so in line with the prion-like spreading hypothesis, here the hypothesis for my project is that alpha-synuclein accumulation leads to brain atrophy and dysfunction by spreading across the brain's connectome, such that the brain's network really shapes the course of the disease progression. And so to do this specifically, I'm using the M83 mouse model. So these are transgenic mice that contain the A53 mutation in the alpha-synuclein gene. And this is a mutation found in one of the familial forms of Parkinson's disease, where the mutation increases the propensity of the protein to form intraprotein interactions, thus leading to a misfolded state, and thus leading to the formation of these toxic fibril forms of the protein, which are found in the Lewy bodies. And so with this, half of the mice were injected with preformed fibrils, so these are synthetically made, and were injected in the right dorsal striatum of the mice. And this is done in order to have a known disease epicenter for which we can map the propagation of alpha-synuclein from that initiation point. And in following the Luke et al. 2012 protocol, all of the mice were sacrificed at 90 days post-injection, and this was a couple days after the mice started developing symptoms, at which point the mice were fully symptomatic. And so in order to examine brain atrophy as the result of the injection of the we acquired ex vivo T1 weighted images, which were acquired at a voxel resolution of 70 microns. And brain atrophy was assessed using a technique called deformation-based morphometry. And so this is a technique that can be used to measure nonlinear local differences in the neuroanatomy. And so the way this works is that all of the mouse brains are registered together through a series of linear and nonlinear registration steps to create a group-wise average. And then each brain is then registered to the average and then back to itself in order to see what changes it had to undergone, which gives us information about the neuroanatomy. So for instance, here we have brain 1, which has a thicker cortex in this region, whereas subject 2 has a thinner cortex. And so by mapping each subject to the average, this area would need to contract, whereas this area would need to expand. And so when we map back from the average to the subject, we get information about volume increases and volume decreases. And so essentially here, where we're getting is a voxel-wise measure of volume difference. And with this, we can then use multivariate techniques in order to examine groups of voxels together. So essentially, we'll be able to get a brain atrophy network. And so specifically here, we use a technique called non-negative matrix factorization, which is a matrix decomposition technique and decomposes an input matrix, which in this case is composed of the voxel-wise volume differences for each of the subjects. And NMF decomposes it into two separate output matrices. The first describing the cluster of voxels in which share a covariance pattern, which can then be mapped onto the mouse brain to see an atrophy network. And the second output matrix describes how each subject loads onto each of these patterns. So here we can assess whether each pattern is more specific to one subject group compared to the other using general linear models. And so here is the spatial pattern of voxels for a significant component that is plotted onto the mouse brain. And this depicts the network of voxels that share a similar variance pattern, where here the color map describes how strongly the voxels covary with the whole pattern. So the more yellow, the stronger the covariance. And so as you can see here, this component reveals a striatal thalamic midbrain pattern, all encapsulating regions that are strongly connected to our injection site, which was the right striatum. So as you can see here, we have the contralateral striatum as well as bilateral pallidum, substantia nigra, midbrain, and PAG regions, just to name a few. And moreover, what we saw was that this pattern of atrophy recapitulates a human PD atrophy pattern, specifically that from Zagami et al. In 2015, in which observed a distribution of atrophy in humans with PD. And these were done using similar techniques such as the T1 weighted images, DBM, and matrix decomposition technique to examine distributions of atrophy. And as you can see here, these two patterns closely mirror each other. Next, we sought to examine the cellular pathology underlying our MRI-derived atrophy results, and this was done using immunohistochemistry, specifically looking at 4 markers of cellular pathology, the first being phosphorylated alpha-synuclein, which is our toxic aggregated form, tyrosine hydroxylase to examine dopaminergic cell loss, EBA1 to examine microglia and neuroinflammation, and finally GFAP to examine astrocytes. And so here is a summary table describing the significant differences in terms of the number of positive cell staining for each of these 4 markers for the regions in which we saw the MRI-derived atrophy. And here, the green p-values reflect higher amounts of cell staining for the mice that received the fibrils, the PFF, compared to the control group, which received the saline injection. And the inverse would be true, which is shown in red. And so if we look specifically at our injection site, we see an increased amount of presence of astrocytes and microglia, as well as phosphorylated alpha-synuclein for the mice that received the fibrils compared to the PBS. But we also saw a significant group by sex interaction, specifically for the staining for phosphorylated alpha-synuclein, where we see males in bloom had a higher presence of the toxic form of the protein compared to the females for the mice that received the fibril injections. And this reflects the sex differences in terms of the disease prevalence, where PD occurs more frequently in men compared with women. And so if we look beyond their injection site, we see an increase in phosphorylated alpha-synuclein staining for the mice that received the fibrils in regions connected to our injection site, such as the frontal cortex, the PAG, and also extending posteriorly to the pons, where we again see this sex by group interaction where the males have a higher presence of the phosphorylated alpha-synuclein. But interestingly, the effects seem to be more pronounced for downstream players of atrophy. So here we see a significant increase in the presence of astrocytes and microglia in the regions where we saw MRI-derived atrophy. And this seems to be a lot more widespread than that of the phosphorylated alpha-synuclein deposition. And so in summary, What we saw was atrophy patterns from mice, which recapitulates human PD atrophy patterns, where we see this widespread bilateral spreading from our injection site to connecting regions. And we saw that the effects of pathology seem to be more pronounced for downstream players of atrophy, as opposed to being a direct effect of the pathological alpha-synuclein at that time point. And so moving forward, we're currently acquiring data to examine alpha-synuclein spreading longitudinally, so specifically looking at time points prior to symptom onset and as well as post once they're fully symptomatic and nearing their endpoint. And this will be done using in vivo MRI imaging. And so with that, I'd like to thank Daniel Galino and Dr. Esther Del Cid Peritolo, in which helped me with the data collection. And I'd also like to thank my supervisor, Dr. Mallar Chakravarty, for all of his help. Thank you.